little bit gears and sort of move from the magic word of evolution to the magic word of ecology. And sort of, I want to start from something that we have. So this is where we are in our plan. And so this is basically what we have discussed so far, right? So this is a measure that you can do in for recent bacteria, and you measure the population abundance and time t, and here you take the log. Okay? So you have your flask, and what you do is you measure how many individuals there are. So this is a proxy of how many individuals there are versus time. Okay? And what you have is that if n of t is e t, then if you take the log, you get something like n of 0 here plus RT. Now don't focus on this part here, and what you see here is that you see something that is a straight line, right? So things grow exponentially. Now of course, not of course. So this is the world where you stay so far. But then if you wait, no. If you wait, what you see is that this doesn't last forever, right? And after some time, you plateau, okay? And the population reaches some value and doesn't move from there. So the question is why?
Not enough space or nutrients?
Yes. So the reason is that exponential growth requires the conditions to be always the same. So it means that if initially there is food for everyone because there are few individuals, then there needs to be food for everyone even when there are many individuals. And you expect that after a while, when the population becomes very large, there is not enough food to sustain the population. Right? There is overcrowding, and then somehow the population cannot grow anymore. Okay? Good. So exponential growth is something that exists only when nothing is changing, and the fact that the population is growing changes something, right? Changes the amount of resources that are available. Okay? So what we want to do now is to, first of all, think about the simplest possible model that describes these dynamics. So and now, in principle, there are many things that can produce this limit to growth. One is, of course, nutrients, resources. There is not enough food for everyone. Another one could be space. If something is space limited, then. But in general, what you can expect is that growth affects the ability of the population to grow. Okay. So let's say that we start from this. Okay. Or actually, let me say, okay, if we start from this, what I can do is that I can take this 1 over n dn dt, okay, on this side, and write this as r. Okay? Written in this way is just to say that this quantity here is the per capita growth rate. Is the growth per individual. And what we have assumed so far is that this quantity is constant. Okay? It's constant, independent of time, independent of everything. The question that I have for you is, if I want to include the fact that resources are limited, and somehow as the population grows, the resources decrease, how should I modify this equation? Okay, so one possibility is that I say that this depends on time. Right? This one is what you were saying? If not,
But it has to be like in terms to the growth rate, because if the rate is over
Okay, so you don't want to put time here. You want to say that this quantity depends on something, right? And what is that something? Huh? For instance, what we can say is that we don't want to measure explicitly resources, but we can say that this quantity here is actually dependent on the population. Okay? So that here we have some function, let's call it g, which now depends on the population. Okay? And so suppose that I want to look at this function g of n. Okay? The population is very small. I can imagine that this quantity here is equal to r. Okay? And then what happens as function of n? What should happen to this function? So I start from here. How should I move my hand? Should go down. Do they keep going down?
It's neutral. It's neutral.
Neutral. So it's like always like this. And then it goes up.
No.
Okay. Okay, what? I agree with you, but I want to understand why you think it should go up.
Why
Because it goes up. Huh?
Why do I think that it shouldn't go up?
Yeah. Or why he thinks it should go up.
Or the times of the population. Now more
You are thinking, yeah, but that should be something that is not a function of the population, right? Then you are thinking that there is a dynamic process that changes the resources and the population, but then it probably is, what you're thinking is that resources arrive again, but then they should be much more, right? Because the population is bigger. So, but let's stick to the simplest scenario, okay? The simplest scenario, I agree that is monotonic, okay? So you think it should be like this? Okay. Okay, so it should touch 0 at some point. Okay. And but what happens? I mean, what happens if I have more individuals than this? Suppose that you are in an environment where you have many more individuals than the food available.
Yes, that would be fine.
Okay.
Also, they should start to go negative.
Okay. So we have something like this. Okay? Good. Now, let's say that we want to write a function that does like that. Okay? What is the simplest possible function you can imagine that does this? Okay? So that starts from here, it's positive in some region, it's negative in other, and touches here. What is the simplest function you can imagine?
R with a line.
A line. Right? The simplest function that I can imagine is a line. Okay? So what is that line? Well, let's write, right? We have 1 over n, n t. What is this line? This line is something that starts in R, right? And then I can say that the point where it touches 0 is some parameter, let's call it k. Okay? So when n is 0, it's here. When it's in k, it's 0. So it should be something like this. Right? So this is a line. This is curved respective line. Okay? So what we have to do is, let's take this simple model and think about what happens. Okay? So now the hard part, we've done it, which is write the model. Then we have just to solve it, right? Which is the easy task in some sense. Okay, so we have this. And so the model we have to solve. Now let me write it in this way. So I put the n on this side, is 1 minus n over t. Okay? So now, how do we solve this model here? The differential equation. It's not particularly hard to solve. We want to solve this with some n0, some initial condition. So we can do it by part to get n1 minus n over k equal to r dt. We integrate on both sides. We can find all of these in the notes. And what we get is, if you take the solution, you get n of t is equal to k, n of 0, k minus n of 0 plus n of 0 into the rt. Okay? So let's look at how this function looks like. So at time 0, where is the population? N0, okay? N0 here. And let's say that the population is below k. We have the other parameter, which is k, which is here. Okay? Let's remember our g of n. So you start in n0, you see that if time is equal to 0, this becomes 1, and k minus n0 to n0 becomes k, so this simplifies to get n0. What if t is very large? What happens to this function if t is very large? So if t is very large, e to the rt goes to infinity, and also these things in the denominator. So these things dominate. So you can simplify this n0 with this one, and what you get is k. So what you see is that this function for large times is to arrive here. Okay? Now this function is to arrive here, and what it does is something like this. Okay? So this term, now if you start above k, what you can see is that you relax. Okay? So with this equation, what happens is that if you start from any positive population and you wait a long enough time, what you expect that you arrive to k, to this value k, which sometimes is called to carrying capacity. Okay? And this equation here is called logistic. Okay? Now, what is the story that this equation is telling us? What we're saying is that if we look at the growth rate, right, the growth rate depends on the population size. And if the population is below k, the population will grow. But somehow the growth rate decreases as the population is increasing because resources are getting depleted. Okay? For instance, or space is getting occupied. So then the growth rate decreases until it becomes 0. Okay? When you are in the carrying capacity. And therefore the population grows and approach this carrying capacity value. If you are above this value, so there are more individuals than resources available for growth, then the population declines until it arrives to this stage. Okay. So this model, even if it is the simplest model, is capturing something fundamental that we were not capturing in exponential growth, which is the fact that when a population is growing, is changing the environment, is changing what's around. So the conditions change because the population is growing. In some sense, exponential growth is the simplest case, it's like the inertial motion, exactly because it's what happens when nothing is happening. Okay? While here, whenever something is happening, the environment is changing, the conditions are changing, and the growth is affected. Okay? So in some sense, ecology, population dynamics, general ecology, is in some sense studying how the growth of populations affect the environment and the environment affect the growth. Right? So this feed. Okay? Which makes it, of course, more difficult and more. Now, there is one thing that, I mean, you can imagine and then you can make all these models more complicated, and we are gonna play with more complicated models today. Before going to that, I want to show you something that you might already know, but that is interesting in terms of how to understand what's the behavior of this model. So in this model, what we did was that we took this equation, okay, and then with all our calculus tools, we solved it and get an equation as function of time, and then we took the limit of large times and we got the solution. Okay, this is what we did. There is actually something that you can do that is, in some sense, much faster and simpler. And what you can say is, suppose that I wait a long time, okay? After a long time, what I expect is that I will be in some value of the population that is not changing, okay? Like here. Okay? And they want to know what is that value. Okay? Now, in order for such a value not to change, okay, what has to happen is that dn dt has to be equal to 0, right? So if dn dt is equal to 0, it means that the population is not changing anymore. Okay? So if I have some dn dt equal to some f of n, okay, I can ask what are the value of n such that this function is equal to 0, right? And if such a value of n is 0, this n star is 0, it means that if I start from that point, the system is not changing. So for this equation, what are the values n star, which corresponds to this condition here? So this n star, this condition here corresponds to these points n star, are called fixed points. Okay? Because those points are the points that don't change during dynamics, are fixed. So what are the fixed points for this dynamics? What I have to solve is R and 1 minus nk equal to 0. So what are the solutions of this equation? 0 and k, right? So there are two solutions. N star equal to 0 and n star equal to k. Okay? So what these two fixed points corresponds to? Well, n star equal to k is what we described earlier, right? So it's if I take a population, this grows, wait a long time, I arrive to a point where there is a balance between the population, the availability of resources, and basically there are as many individuals as the environment can sustain. Okay? Because one more individuals will be too much, one less individuals will grow. Okay? So this is what we described. What is n star equal to 0? Why this is a fixed point? Okay? What does it mean that n star equal to 0 is a fixed point? Yes, because there is no spontaneous generation, right? Because if we have 0 individuals, at least in this model, there are 0 individuals forever. Okay? Right? Now there is something different between these two fixed points. Okay? There is something fundamentally different between them. What is the fundamental difference? Let's imagine that you take these fixed points and you make a small perturbation. Okay? So you are at k, you have k individuals, and then you say, now I have k plus 1. Or let's say that you are at 0, and now you say you are at 0 plus 1. What's the difference between these two points?
The stability.
The stability, okay. What does it mean, the stability?
That if you're in 0 and you add 1, it will grow and not
Exactly.
return to 0.
Exactly.
But if you are in k and you add a perturbation, you return to k.
Exactly, exactly. So the point is that if we are in 0, if you are in 0, say that you're here, if you add 1 individual here, if you start from an initial condition which is slightly above this value, right, you don't go back, you actually grow, you go away from 0. Right? While if you start exactly above k, you go back to the value. Okay? So in some sense, we see as a notion that this is, it's called stable. If you want to locally asymptotically stable, if you want the right jargon, which means that if you perturb it, you go back to it. Well, this point is unstable. Okay? If you perturb it, you go away. Okay? Is it clear? Good. Good. So this is the simplest possible model that captures a fundamental process of ecology, which is competition between conspecifics, right? So if you have a population of individuals that are all identical to each other, they will not grow forever because resources are limited. They compete for these resources, and this limits population. Okay. Now, this is sort of the starting point, and you can imagine that now a new world opens, right? Where you take a single population and you can imagine that these individuals interact in more complicated way. They can help each other, for instance, or they can do other stuff. And then you can also say, okay, now I don't have just one population. Okay, perhaps I have multiple populations. So I have species of one tree or another tree, another three species, and they compete, but they compete in different ways, right? If an environment there are many individuals like me, or many individuals like a different species, the effect on each other is different. Okay? You can imagine of building more and more complicated models. But what I want to do is to focus on another type of interaction, which is a sort of the most iconic ecological interactions, in the sense that it's the one that you see in all documentaries that you watch on TVs, right? And what is that ecological interaction?
Predation.
Predation, right? I mean, there are many ecological interactions that are very interesting, like competition. But you can, I mean, it's beautiful to look outside. We can see that these trees are competing, but I mean, there is not much to see about that competition, right? When predation is definitely more, there is more action. Okay. Okay, so let's look at competition. And what you can do in competition is that now you have two different species. Right? You have a population of predators, right? And you have a population of prey. And what you can do is that, let's say you have prey and predators. And you can look at how these two quantities are changing over time. Okay? Right? So you can have some dynamics about predators and prey. Okay? Now I'll show you one. There are many things that can happen. I'll show you one interesting thing. Okay? One interesting thing that happens is that, so these are data, okay, of three very different systems. Okay? So this is lynx and hares. This is rotifers and algae. These are bacteria and phages. Okay? So you see that the times here is tens of years. This is in days. This is in hours. Okay. Here you have thousands of individuals. Here you have some number you have 2 to the 10 to the 5. Here you have 4. You have 10 to the 4 individuals per milliliter. But what you see is that you have oscillations. Okay, things go up and down. Okay, you have things go up and down. Here actually is interesting. Go up and down and then stop. One stop and the other one go up and down. Okay, this is an interesting story. But at least if you look at these one here, they both go up and down. Okay, So what we want to do now is to study.
That's a bacterial resistance.
It's interesting. It's a cryptic cycle. It's called. So basically, here, but if I remember, but this is driven by evolution. You have different populations. I think of bacteria and phages that are co-evolving, and so the result in this. But okay, another story. We will see something.
So what was the name of the equation you showed before? The
Logistic equation.
Logistic.
Logistic growth. Okay. So what we're going to do now is to write a model to describe these kind of cycles using these as an example. Okay. And so we're going to now describe the ecological dynamics. And then next week, we're going to start thinking about eco-evolutionary dynamics. So how does a population of phage and bacteria, or just phages, evolve in an ecological setting? Okay? When there is not an abstract concept of fitness, but there are different traits. And these different traits can evolve. Okay? So for the rest of the time that we have together, we're going to think about these two things. Okay? But many of the things that we say apply with slight differences to other systems. Okay? Now also realize that one interesting fact about this type of predation is that in this case, the predators is bigger than the prey. While in this case, there's slightly a different story in that respect. Okay? Anyway. Okay, so let's go back to the system that we're going to study. So what we're going to study is these things that you already see. Right? So we have bacteria. This bacteria can be infected by, a phage can inject their genetic material. The genetic material contributes to the production of new phages, and then the bacteria dies. Okay? So what we want to do is to describe a model with differential equations like the ones we wrote before that describes these dynamics. Okay? And we're going to do it together like before. So let's start from this picture. And let's think about what are the variables that we want. Right? We want to write some, like earlier we wrote dn dt equal to something. Right? Now we want some d dt equal to something, and they want to first think about what is this thing in parentheses. Okay? What are the variables we are describing? It is not only one variable, there are multiple populations. So what are the variables? Yes, so I have I have one population of bacteria. So let's say I count the bacteria, N. Then what I have to count? The phages, so V, viruses. That's it. Huh? I don't think so, because otherwise I would not say it. So we have the population of bacteria. Now, I have the population of bacteria that are happily there. I have the population of phages which are happily there. Okay. What is missing in this figure? Well, the bacteria are here, N. Okay. The infected ones, right? So I have the infected bacteria. Okay. So let's use capital letters. Okay. So you have the bacteria infected cells, and then I have viruses. Okay. So these are the I could think of more, but this is the thing I want to write the equation. So I will have some dn. I will have some dn t dv and di dt and some dv dt. Okay? Now, let's start from the things we know. Okay? Let's imagine, first of all, that there are no phages. Okay? If there are no phages, what equation should I write for the NDP? Okay, I can say it's Rn, but then we discover something that is important about this population, which is yes. So I will start by writing this. Okay. Now, if there are no phages, this is the only one. This is everything they need. Right? That's it. This is what we started from earlier. Now I want to add phages. Okay. Suppose that I want to add phages. How should I add them? What do phages do? Infect. Okay. So what I have to explain first is this error here. Okay. So this error here is something that transforms an happy cell in an infected cell. Okay. So I have a term here with a minus, which corresponds here to a plus. Okay. Because I move something from, I remove some cells that are naive, and I create some cells that are infected. So now what I have to define is at what rate this happens. Okay? So at what rate do I do a virus infect a cell? Okay? Well, there will be some parameter, okay, for sure. Let's call it phi. Okay. Because we like Greek letters. There is some parameter, and this is something that tells us how fast things happen. Now, what I have to specify is how these infections depend on how many bacteria I have and how many phages I have. Okay? So if there are more bacteria, there will be more or less infections? More, right? And in general, what we can expect is that the number of infection grows linearly with the number of bacteria. And okay. And then if I keep the number of bacteria fixed and I have more phages, do I have more or less infection? More. Right? There are more phages, so I have V. Okay. So if you want, N times V is like something that captures how often a phage encounters a bacteria. Okay, so this is about infection. Now, what I have to add is something about the second error. So if you want, we can write the first error here. It's phi and v. Okay. Now there is this second error here. Okay. So what determines the transition from this point to this point? Speak louder. Huh?
We cannot hear you.
Yeah, well, it's lysis, so it's like basically what is the process that have to occur from infection to this point here? Yeah. Is like the fact that viruses have to assemble, right? So they sort of, there is a process which is sort of biological, and it takes some time. So that process takes some time, and that time does not depend on, let's say, how, if I take one infected cell, how much it takes, it does not depend on how many other cells there are around. Right? So let's say that this rate here, it's eta, okay? So if you want, this is a rate, how fast is the process? So the larger is eta, the faster is the process. If you want 1 over eta is the time it takes, okay? So this means that with some rate, minus eta, okay, a cell stopped to be infected. Now this eta is for every single cell, so if I have more infected cells, there will be more cells that will stop to be infected. Okay? And then when a cell stops to be infected, lies, right? And then boom, okay? It really explodes and release viruses. So here I have beta I. Now, how many viruses does it release? Some number, okay? It's a parameter. So this number here, how many I have, it's called the burst size. Let's call it beta, okay? So this is beta I, okay? Now I want to add one other term, which is the fact that I say, suppose that there are no cells around, okay? There are only phages. These phages will not stay there forever, right? Because they decay or they are diluted or whatever. So there is some mortality of the phages. And so let's call it minus omega. Okay, omega p, sorry.
You also assume all of the cells that die are because of infection.
Sorry?
You also assume that all the cells that die are dying by infection.
Yes. I mean, in principle, you could add another term, which is here minus dn, and also here, also here you could add it. And this minus dn, it can be reabsorbed in this R, right? Because it has the same, so how it's. Good. So we have, I think, our model, right? Is there anything I'm missing? No. Ah, yes, there is one thing that I'm missing. There is one thing that I'm missing, which is every time I have an infection, there is one less virus around. Right? So every time I have an infection, I should divide the viruses that are around, decrease by 1. Right? Do you agree? So when there is an infection event, right, I increase the number of cells by 1, I decrease the number of viruses by 1, and I increase the number of infected cells. Okay? Do we miss anything?
The omega b, what did it do?
Sorry?
The omega b, the
This is like some sort of mortality of viruses. Okay? I mean, in principle, I could add mortality here, mortality also here, but it will be effectively reabsorbed. In principle, I mean, one can do it, right? You can say, okay, here I have minus d and minus di. Okay? I'm not doing it because I'm thinking that the main, this term here is sort of here because it's inside R. R is a birth minus a death rate, no problem. This term here, it's actually not important because you say these are like bacteria that die anyway because of viruses. And it's way more likely that when they are infected that they die because there are viruses than other processes. In the next part, we're going to add this term because we want to say something. Okay? Okay. But for now, let's stick to it because it's simpler. I want to say, well, this term is important, right? This term is saying if there are no bacteria around, so n is equal to 0, I is equal to 0, the viruses are decayed. But this has to be negative, right? Because in absence of them, they don't grow. Okay? Good. So, if this is clear, now we can forget about biology for a second. I mean, the reason why we write these things is because we thought about biology, we wrote this. Now what we have to do is just to follow the math. We follow the math and then we go back to biology. Okay? So now it starts the easy part. Okay? We did the hard part. Okay? I'm serious, eh? I know that sounds counterintuitive, but it's pretty easy. Okay? Because now these things you can ask ChatGPT, please solve this equation. Okay, it will solve it. Right? You ask a mathematician and you say, solve this, it's gonna solve it. Right? But going from this to that is much harder. Okay, so we have the model. So now, how do we go about doing the easy thing? Okay? What we need to do is to solve this model. What does it mean to solve this model? Well, one thing you can do is that you can write a program that simulates these dynamics. Okay? So what you do is that you have a numerical integrator that integrates these dynamics. Okay? So you start with some initial conditions and you integrate. Okay? The code you have access to does exactly this. So what I'll show you is the outcome of this, but you should play and explore what are the results. Okay? And now let's try to understand a little bit how to get an intuition of what's happening. So now, what time is it?
17.
So you have 30 minutes.
Huh?
I have 30 minutes.
You can take.
14. I have 14 minutes.
You can take a little more. We're ready for lunch. Yesterday they came like 10 minutes late or something.
Okay, that's good. So we saw in logistic growth that we can solve exactly the equation. Now here, you cannot even dream of solving this exactly. But we also saw in logistic growth that if we want to understand what happens over long times, we really don't need to solve exactly the equation, right? We can first of all look for fixed points. Okay. So how do we look for fixed points here? Yes, we said to zero what? Which equation?
Whatever.
All of them. This is important, right? You want to fix all of them being equal to zero. Because it's not that you don't want n to change. You don't want to n, I, and v to change. Because if they change, they're going to affect each other. So I want to set these equal to 0, equal to 0, equal to 0. Okay? So let's do like, all the calculation is done over there, you can read it if you want. But let's look at the key points here. Okay? So let's look at the first equation. The first equation is proportional to n and to n. Okay? This is an important feature of ecology. So I can write it as n r, sorry, that's r1 minus n over k minus b equal to 0. Okay? Yes?
Parameter missing.
Yeah. Okay? Thank you. Then I have the second equation, which is, I can write it simply as P and B equal to eta I. Eta I equal to P and B. What this equation is telling me, telling us, is that at equilibrium, to be in equilibrium, there must be a balance between how many new cells are infected and how many cells infected cell lies. Okay? So this is that balance. And then if you take the third equation, what you get is beta I minus P and B equal omega v. Okay? But then, if you look at this equation here, or actually, let me write it like this, minus omega v. Now I can take this second equation because I know that eta I is equal to phi and v, right? So this term here, I can put it here, and this is exactly equal to this. So I get beta minus 1 phi and v minus omega v equal to 0. Okay? Which this is proportional to v and to v, and what I get is this one minus omega v equal to 0. Okay? Good. So what we have to solve is now this equation we already used it. So we have to solve this equation here and this equation here. Okay? Which is relating how many naive bacteria, not infected bacteria we have, and how many phages we have. Okay? Now, if you look at this equation, how can you set, let's say, let's start from this equation here. How can you set this equation equal to 0? There are two ways in this equation can be equal to 0. One way is if v is equal to 0, and the other way is if n is equal to 0. Okay? So, one case is that if we look at n star and v star, we have one scenario where v is equal to 0, and then v is equal to 0. This is what we started from or not? Yeah, v equal to 0, then you don't have this term. So you have two ways on making this equation equal to 0. Right? It can either be n equal to 0 or n equal to k. Right? So you have 0 or you have k. Right?
There is another one, right?
There is another. There are more than one. There are other two. Okay? Right. Then, okay, suppose that, well, there is another one. Suppose that v is equal to 0. Okay? Is different from 0, sorry. If v is different from 0, this term has to be equal to 0. Okay? So we get that n star is equal to beta minus 1 omega over phi, right? Because v is different from 0. I get this equation here. And then if I substitute this value of n here, I get some v, which I will not run. Is this clear, the structure? So basically, I have these two equations, right? So I can make them equal to 0 by setting this 0 and this 0. First one. Then I can set this 0 and this 0. Or what I can do is that I can set this term is 0 and this term is 0. Yes?
Shouldn't the term b minus beta minus 1 should be divided for?
Yes, sorry.
Okay. Yeah.
Okay. Now, what are these three states? What do they represent? So, this state here represent the case where, what is that state? Well, they are all fixed points, but what does it mean, 0, 0? There is nothing to look, I mean, we are doing physics, not biology, okay? So there is no life. So this is the uninteresting state, okay? So no life. What is this state?
Where there's no virus.
No phage, exactly, no phage. What is the state? Coexistence. Okay? Okay, so this is good. So I want to comment something which is very interesting. It's written there, but it's not immediate if you don't really think about it. So here, when you don't have phages, okay, what sets the population abundance of the bacteria? When there are no phages, what is determining the population abundance of bacteria? K, the resources. What is determining the population abundance is K. So how many resources there are, right? Exactly. So depends how many. Suppose that I have this culture of bacteria and phages. If there are no phages, it depends how many resources. Now, when there are phages, what is determining the population abundance of bacteria? What are these numbers?
The decay of bacteria.
No, the decay of phages,
Yeah. Sorry.
the infection rate of phages, the birth size of phages. So what sets the population abundance of bacteria is not the properties of bacteria, are the properties of phages. Okay? And vice versa, if you look at this, at v tilde, now let's solve it. V tilde is equal to, so if I set this equal to 0, is R over phi 1 minus N, which is given by this, over K. So omega beta minus 1 phi times K. Okay? So what sets the population of bacteria are purely the properties, the traits of phages. If I put more resources, I will not increase the population of bacteria. I will increase the population of phages. Right? It is very counterintuitive, okay? But it's a fundamental process of predation, let's say, in ecological system. That when you have like predation, you have something that is called top-down control. So the predator controls the population of prey. And if you increase how many resources you have, you are not increasing the population of the prey. You are increasing the population of the predators. Okay? And so this is why, for instance, is extremely important. I mean, now not thinking about phages and bacteria, but if you think about fishery, so or like, yes, fishery, it's extremely important the role of predators in an ecosystem, because predators are controlling the biomass of prey. Right? And if you kill them, you can completely change the population of prey. Okay? So these are what we have as fixed point. Now, are we satisfied with this, or do we have to look at something about that? Well, we don't want to solve the equations because we are lazy and because we have to go to lunch in a few minutes. So what are the properties we care about? Whether these points are stable. Stable. Okay. What we care about is whether they are stable. Now, I don't have the time in these days together to cover how you check stability. But in some sense, what you do is that you ask something mathematically, it's something that is very mechanical in some sense. And the idea is you assume that there is a small perturbation around this point, okay? And you ask whether this perturbation goes back to the initial state or moves away, okay? So you have these techniques where you can apply it very easily. What I want to do now is to think about these two fixed points here, okay? And think about how you transition, no, sorry, not these two, this is 3 here. These two fixed points here, okay? And I want to ask how you go, under what condition you go between one and the other, Okay? So, suppose that you are here, okay? Now let's build an intuition about what this stability means. Suppose that I'm here, okay? I have K bacteria that are living happily, carrying capacity, and then I add a small amount of phages, okay? How do I know if I'm here or if I'm there? What should I measure? Suppose that you have this bacterial culture, I have K individuals, I add a small amount of phages. What do I expect? That if I'm here, the phage will go to 0. If they're here, they will increase. Okay? What is determining the increase of phages? The derivative. Right? The derivative of the phages. So what I can say is, suppose that you are so suppose that I have exactly K bacteria, okay? I have exactly K bacteria because I'm here, and so N is equal to K, okay? And then I add a small amount of phages, which is very small, okay? So if I add a small amount of phages, what is going to happen is that, well, what I can say is how many infected cells I will have will be this number here, okay? Which is very small. So they are infected, and I will have this number. Okay? So what I can say is that I will be small and will be equal to phi n v over t. Okay? And so with this small v, what I can calculate is what is the vdt? Well, I will have to substitute it here, and I get that this is beta minus 1 times phi n v, okay, minus omega v. And so then, if I look at this equation, this will be equal to v beta minus 1 phi n minus omega. Okay? And so when will the phage grow? If this term is positive, this is the growth rate of phages, right? This n, I can imagine that this n is equal to the carrying capacity, so this is K. So initially, the phage will grow if this term is positive, okay? And will not grow if this term is negative, okay? So I can say that this no phage thing, okay? I will see no phages if they don't grow, right? So if this term is positive, it's negative. So if beta minus 1 phi k minus omega is negative, okay? Which means that beta is lower than 1 plus omega over phi k. Okay? So if this beta is small enough, phages are just not able to grow, right? Because when they infect bacteria, there are more phages that die than the ones that can infect bacteria. Okay? If beta is larger than this value, then bacteria can invade. Okay? Now, this is not the end of the story, because this is telling us under what conditions this is not stable. What you should ask is whether this is stable or not. Okay? whether you stay there forever or you have fluctuation, or you sort of go away. And if you do all the calculations, what you discover is that this quantity here, this fixed point here, is stable if beta is between that value. So if beta is larger than that value, but also if beta is lower than some critical value. Okay? And so, just to conclude, okay, I'll show you what happens here. And what we sort of, I introduced to how to, I didn't do the calculation, but I introduced to what is the way. The result is that, well, this state is never stable, so this one is, we don't care, okay? So we have scenario 1, no phages. If beta is lower than 1 plus phi, plus omega. Okay. Then we have scenario 2, fixed point, coexistence. If beta is larger than 1 plus omega pk and lower than beta c. And then we have scenario 3, which is beta larger than beta c, but we have to see what happens. Okay? So these are the 3 scenarios. So now, if you go to the simulations, you can play with it. And so, let's erase this. And so you can simulate. What do you do when you simulate? Well, you specify some initial condition, and then you numerically integrate the dynamics, and you look at trajectories over time. Okay? I think you have to click. Yes. So this is the scenario 1, right? When beta is smaller, okay, smaller than some values, if we start from 1, so here I start the simulation from exactly k bacteria. Okay. This is k. Okay. You start with 1 virus. Okay. And then you look at the dynamics. Okay. You look at the dynamics, and what you see is that the bacteria, the viruses, well, here you have an infection actually, but then they decline. Now, here 1, it's concentration, so you can have more. But basically, they decline. Right? This is a log population. So what it's telling us is that this is declining at a fixed rate. It's going to extinction. Okay? We actually calculated what is this rate. This rate would be beta minus 1 phi k minus omega. Okay? So they're going to extinction, which is a negative number in this case, because beta is small, so they're going to extinction. And the number of infected cells is also going down. Okay? Because you have, so in this case is no bacteria. Now, what if you have intermediate situations where you expect coexistence? So you start from the same initial conditions. Now, so they were not aligned. So this is again 1, and this is again k. And what happens? It happens that bacteria infect, right? The number of infected cells increases, release more virus, increases, and the number of bacteria decreases, and then these things balance. Right? When you are in this case, basically bacteria infect this, and the rate is equal to the rate of new viruses, the rate of decrease, everything is balanced. Okay? So then what you can do is, let's increase beta even more. Right? Increase beta even more, and then what happens is, what do we expect it happens? Huh? Well, if the bacteria die, then, a good point, but if the bacteria die, then also the viruses have to die. Right? So let's say that the bacteria die. That's an interesting point. Let's say the bacteria die. So then there are no any more bacteria. So what happens to the virus? They die. But then what happens to the bacteria? They grow again. And then what happens to the viruses? And then what happens to the bacteria? Then what happens to us if we keep doing this? We don't go to lunch, so let's stop. So what you see is cycles. Okay? So you see that what happens is that bacteria phage infect. Now they produce many more phages, so they drive the bacteria to very low abundance. As you say, bacteria die. But then when you are at this point, there is no more food there is no more opportunities for phages. So they decline. And as they decline, the population of bacteria increases. Right? But then there is more opportunities for them, so they grow. But then they kill them. Okay? And so what you have is that you have a cycle. Now this cycle is very interesting. It's called, it's something that you don't see in physics. These are called the Grilli cycles. Which means that these are cycles where the amplitude does not depend on initial conditions. In physics, you see oscillations all the time, the pendulum, right? But this pendulum, it's not the oscillations like this. Because in a pendulum, if I change the initial condition, I have larger oscillations. This is something where the oscillations are independent. Okay? And so what we see is that we see these cycles, right? And so this sort of explains the initial observations that we had with the different predator-prey systems and oscillations. Okay? I think it's time for us to be in this phase. Okay? Increase. So questions? Good. So next week, what we're going to do is that we're going to think of what happens if now you add evolution to this. Okay? Good. Thank you.
Oh, I guess.